Yeah. But then the inside has to be cooked.
My dad was a really good
Turn up a little bit.
He always marinate food out.
All right, everybody. Thank you all for attending our undergraduate showcase. We are going to get started right at noon because we have a lot of speakers and we want to make sure everyone has time to get through all of their talk, maybe answer a couple questions. But thank you to all of our undergrads who agreed to come share their research. You guys do a lot of great work in our department, in our lab. We're really excited to hear about all the stuff you've done. So it's going to be pretty quick. Everyone's going to give their talk. We're going to have time for like 1 or 2 questions while the next person rotates up, and then we're going to move on. But yeah, let's go ahead and get started with Mark. Thank you.
Is this thing working? All right, cool. All right. So as previously stated, I'm Mark, in case you forgot. I'm from the Wittkopp lab. I'm here to talk to you about the association of bacterial enrichment with pigmentation variation in Drosophila americana. Kind of a mouthful, a lot of buzzwords in there. Let's kind of unpack this. Why are we doing this in Drosophila? It's the most annoying insect known to mankind. So Drosophila are actually one of the greatest genetic models you can kind of look at. So you can generate a lot of them super quickly. They've got a super short life cycle. And because of that, it bleeds into the next point. If you squish 1, if you squish 10, you've got 50 more ready to go. They're kind of a hardy insect. You can dissect them, move them around. Like they're not just going to disintegrate on you. They've been used a lot in the past as genetic model. So there's a whole lot of resources out there that are available for you to work with. But the most important part of why they're a genetic model is there's a lot of traits that have been genetically mapped. And because of that, they're very easy to track through the different generations. So one of the things that we're interested in as a lab is the pigmentation variation as a model trait. So go with this image. As you can see, just within species, there's a whole lot of different kinds of wings. There's a whole lot. Some of them are striped. Some of the legs are striped. Some of them have dots. But there's just a whole different amounts of pigmentation variation. And I'll use this to go through the incredibly complicated anatomy real quick so you know which terms I'm using. Do I have a pointer? Thank you. Oh, that would be helpful, wouldn't it? Cool. All right. So you've got the head. You've got one of those. It should be pretty easy. You've got the thorax and the abdomen. That's it. All right. Cool. So we're mainly interested in the abdominal with pigmentation, which have been spread open for your viewing pleasure. But as you can see, there's just so many different kinds of, when I keep saying the word variation, but there's so many different kinds of variation within just the species. As you can see there, there's some in the head and the abdomen and the wings, but we're mainly interested in the abdomen. So some of the things that I've done to kind of further this information are a technique called HCR. HCR essentially allows you to see the gene development during the gene expression during development. Sorry. I've done this in 3 different species. This is melanogaster because it looked the prettiest. But I've also done it in americana flies and Nova mexicana flies. So the thing over here, one of the things I'm bringing up about why this is so unique and I find it so fascinating is you have over here, this is called the tan gene. You don't need to remember that. But if you see here, there's a lot of red that is consolidated on the bottom. And then if you look at the abdomen of a complete and adult fly, they have almost the same kind of pigmentation going on. One of the other things I've kind of done is, and a lot of other people have done in the lab, is just some crossings between some light and some dark flies, observing the phenotypes, some pretty basic, it's pretty basic cross. So, but how does any of that tie into the bacterial part? Because I've talked a lot about the Drosophila part. How does the bacteria come in? And that's where we know that bacteria influences organismal traits. So something like the bobtail squid uses bacteria to hide in the moonlight from predators through its bioluminescence. You have one of these, so digestive system, you should be familiar. You absorb nutrients with the bacteria in there that helps break down food. It's important for your development. But we've also found in melanogaster that it's linked to their sexual reproduction and their general life cycle. So there's a postdoc in our lab, Lena, who had noticed while working with americana flies that there had been some kind of, she's pictured here, had been noticed, or she had noticed that there was a higher bacterial load on the lighter pigmented flies and a lower amount of pigmentation on the darker. So that caused some more research into what stuff has been done prior. And there's not a whole lot, believe it or not. No one's really looking at these things microbiome too extensively. And what we had seen shows a loose link, but not as specific as we're hoping for. So we're doing this project. So I'll introduce you, the star of the show, the Americana. I've killed probably 100 of these things since I've started. Actually, hundreds, sorry. All right, so this is the thing. Here's the fly itself. Americana are a lot darker than melanogaster. They're really fat. When they come out, you have to like not pop them, which I've done a couple of times. It's not fun. But they also they have a lot longer of a life cycle. But the most important part for this project is the fact that you have all of this variation in the abdomens. And that kind of allows us that gives us a tool in order to analyze what we're going after. So how do you quantify something like this? And we've been conducting an experiment that looks similar to, or just like this. We're gonna take our fly. Please do not get attached to him. He is not long for this world. We're gonna give him a nice bath before he goes. And then we have a split in the path. We're gonna take his bath water and we're gonna dilute that, plate that, incubate it. And then the fly himself is gonna get 2 more baths before, if you've ever worked in a restaurant like me, the next step's a little cathartic because he's gonna get crushed up, diluted, and then that liquid will get plated, incubated, and we're counting up the colonies of the surface and the gut bacteria to see the different correlations. So what does an example of my data look like? We've got 2 different plates here. One of them has 192 colonies, the other is 226. The 226 is for the light, the other one's for the dark. That is a connection to what we've been saying, where the darker has a lot more bacteria than the light does. Or not, reverse that. The light has a lot more than the darker. So where do we plan to take this further? We've got, first of all, I've got about 70 images of gut bacteria that I would love to analyze the second organic chemistry isn't ruining my life. And I'd also, since both of these two that I mentioned earlier are kind of 2 extreme phenotypes, this one's super dark and this one is known for being super light, I'd like to fill in the middle here. It's a pretty sizable gap. So that way we can properly see if there's a regular correlation and a regular slope, or if not. I'd like to give some quick acknowledgements. Thank you to the Beckman Scholars Program for providing funding for the lab, myself, my PI, keeps food on the table for me at least. I'm very thankful for the opportunities that they have given me. My PI, Trisha Wittkopp, for giving me the space to pursue research as an undergrad and trusting me around stuff worth way more than I will ever earn. And then some key lab members, in particular, Eric and Teslima. Teslima took me on as, or she's my mentor. She took me on as a protege of sorts. They've trained me. They've shown me how to do the different techniques. They've worked with me on experimental design and also dumbing down papers so I can understand them. And also, she's not here, but Rebecca for memorizing my entire calendar and making sure I never miss a meeting. Never asked her to do that, but I appreciate it. Questions?
Any questions for Mark?
Bacterial genera are fascinating because they are involved with the substances these flies like to hang around. And so I wonder how much you've investigated the nature of the bacteria themselves, Acetobacter and Lactobacillus.
So are you asking like the bacteria that's like native to the fly?
Yeah.
That's kind of like the part of this. I haven't necessarily looked into the nature of how they operate yet, but at least that's the point of washing them, to get rid of anything else that they may have come in contact with in the lab and all the other flies that they interact with in their tube.
Yeah.
But I don't have the most data about how, like, the nature of the bacteria, other than the fact that we have a link between the amount and how dark and light they are.
Right. And so the ones that you're analyzing in your colonies, are those ones that you think are coming out of the gut or on the skin or both?
Both.
Or can't you tell? Okay.
Yes.
We have time for one more question.
Have you thought about looking at the larvae to see if they're influencing the earlier stages?
Looking at the larvae to see earlier.
Yeah,
Yeah.
whether they're associated in that stage.
I haven't, but I think that's a good idea. And I will start looking, I will, at least that's an avenue to explore.
I did appreciate organic chemistry much later.
I don't appreciate it right now, so I'm hoping I can get to your point.
Mark. Perfect.
Go, Lena.
Hello, everybody. My name is Lena Reeves. I'm an UROP student in the Classen lab working with audio mostly in the cloud. And today I'm going to talk to you about the effects of Bracken fern on traits and germination of Acer rubrum seedlings. So to get started, I'm going to take you all to the University of Michigan Biological Station. At UMBS, the forests have their understory is dominated by Bracken fern. It is quite literally everywhere. But the really cool thing about Bracken fern as a dominant species is that it is also dominant across the globe. And so that really led us to start to wonder what its role in the greater ecosystem was. And there's also a really large body of literature that looks at the effects dominant species have on their ecosystems. And one of the key ideas of this literature is the mass ratio hypothesis, which essentially states that a species biomass within the community or their dominance within the community has an effect proportional on that ecosystem. So as you can see on the y-axis, as a species is more abundant within the community, they're more dominant within the community. And this was introduced in 1998 by ecologist J.P. Grime. And with this mass ratio hypothesis, we thought to ask our question, which is, what effects does the dominant understory species, Bracken fern, have on plant traits of red maple seedlings at the University of Michigan Biological Station? So to look at this, we came up with two hypotheses. Our first hypothesis was the light competition hypothesis, which is that Bracken fern is inhibiting light from reaching other species in the understory, and thus that is why other species are not growing, and that is how it is affecting its ecosystem. And then the other hypothesis was the chemistry hypothesis, which is that there is some other soil-mediated process happening that is keeping other species from growing within the ecosystem. For the light competition hypothesis, we hypothesized that in natural plots, as Bracken coverage increased, other biotic and abiotic factors would decrease in the plots. And then when Bracken was removed, the abiotic and biotic factors would stay consistent across the plot gradient. However, with the chemistry hypothesis, we hypothesized that in both natural and removal plots, as the coverage increased of Bracken fern, the abiotic and biotic factors would decrease because it wasn't something like Bracken fern blocking the light, but there was something actually happening in the soil that was inhibiting growth. To do this, we set up observational and removal Bracken fern plots. We had 30 plots, half of which we removed all of the Bracken fern in, and the other half we kept it at its natural gradient. And it was at a gradient of 0 to 100% Bracken fern. And with this, we planted our Acer rubrum seedlings at the beginning of the season, observed them throughout the growing season, and then removed them at the end of the season to look at functional traits. The main traits that I looked at last year were specific root length, specific root area, and specific leaf area. And these are all traits that are really important for nutrient acquisition in Acer rubrum seedlings. And what we found was really interesting. There was no significant difference within or between plots of natural and removal Bracken fern coverage for any of these functional traits. So that led us to sort of move on from the light competition hypothesis and move to the chemistry hypothesis. And what we are thinking currently with the chemistry hypothesis is that maybe Bracken fern is allelopathic and inhibiting growth through allelopathic chemistry. And this has also been something that has been studied. In California in 1976, ecologist Gleasman did this study where he planted all of these species in plots with bracken fern and in plots with grass. And as you can see, the plots with bracken fern had significantly lower dry weight than the plots with grass. And he also repeated the study in Oregon and found very similar results, where plots with bracken fern had little to no dry weight versus the control plots, which had a significant amount more of dry weight with other species growing in them. So with this, it led us to ask the question, does bracken fern allelopathy have an effect on red maple Acer rubrum seed germination? To do this, we conducted a bioassay and we stratified our seeds. We did this to emulate winter. It's 30 days in the dark, in the cold. And then after these 30 days, we transferred our stratified seeds into Petri dishes. Each Petri dish had 22 stratified seeds, and there are 25 dishes and 5 treatments per, 5 treatments and then 5 plates per treatment. Our treatments are a cereal dilution of bracken fern leachate. So essentially, we made bracken fern tea, and we put dried bracken fern into 1,000 milliliters of water at their percentage weight for 48 hours, and then we drained it, and we are using that to water these Petri dishes over 28 days. So these are our 5 concentrations. We have a control, which is just regular water, 5% dilution, 3.75%, 2.5%, and 1.25% dilution. And to conduct our bioassay, our Petri dishes are in a growth chamber for 28 days. For the first 14 days, we have been watering them every day with 10 milliliters of their respective dilution. And then for the second 14 days, we're watering them every other day. And we're really looking to see how many seeds actually germinate when in contact with this bracken fern leachate. And so our predicted results is that in the control plates with 0% leachate concentration, that about all of the seeds should be germinating because they're not coming into contact with this bracken fern. And then in the plates with the 5% dilution, we are expecting that none of them will germinate. And so, oh, whoopsies, that is our project. Thank you so much.
Questions?
Well, I can't wait to see what you find.
Yeah.
Sometimes one of the hardest parts of experiments like this is just figuring out like what your treatments should be. How did you decide what percentages of leachate to use?
Yeah.
Because I imagine it's hard to figure out what the seedlings might be experiencing.
Yeah. We did a lot of the Gleasman paper was looking at like when in the season the leachate was actually affecting the plants. And they found that an ecosystem similar to Michigan, it was like post-winter after the first snow melt. And so we were trying to take that into consideration with the stratification process. And then also we just did a lot of paper reading, and a lot of the cereal dilutions were for bracken fern around the same, like 5% to 1.25%.
We have time for one more.
What happens if it's all the same?
If it's all the same,
Then what? What's next? What do you do if it's all the same?
I think the next step would be to look at if it's not germination that is being affected by the bracken fern allelopathy, maybe it's during seedling establishment that these seeds are and these plants are being affected by bracken fern.
So you're going to stay with us for like the next couple of years.
Sure.
To figure this out.
Yeah. Yeah.
All right. Thank you to Lena. Samantha, you're up next.
Is this okay? Okay. Hi. Oh, wait. Hi, I'm Samantha Molino. I'm in the Weber lab, and today I'm going to be presenting on the research I've been doing for my senior thesis on ferns also, Domatia, and the evolutionary convergence of a mutualism. So I first want to start off by kind of framing my talk around the multitude of mutualistic relationships that plants engage in. Land plants have this rich evolutionary ecology, and as such have a lot of these iconic examples of mutualisms that I'm sure we are all aware of, often exchanging nutrients for a reproductive or defensive benefit. I've included some examples here, but you have the plant-pollinator relationship, the nectary bodyguard arthropod relationship, and the root system mycorrhizal fungi relationship. These are all really common, really well known, but there is another mutualism that, despite being really widespread, you might not know that much about, and that is Domatia. So Domatia are these structures that are made up of trichomes, and trichomes are these little kind of plant hairs that cluster together in these tufts, and they're found on the abaxial or underside of the leaf in between the mid-veins. And these structures actually help facilitate a mutualistic relationship between the plant and beneficial mites. So the way this works is that the plants provide shelter for the mites, so the mites can live in these structures and lay eggs there, and because they're on the underside of the leaf, they gain protection from UV, desiccation, and rain. And then in turn, the mites provide protection as they are predaceous and fungivorous, so they will clear the leaf of smaller herbivorous arthropods, often smaller mites, and fungal pathogens. Now, Domatia are actually really widespread. They're found in over 2,500 angiosperm species, and there tends to be a global trend of them being more common in temperate regions. Now, angiosperms have undergone over 130 million years of selective pressures, and the multitude of independently evolving lineages that have Domatia indicate that this is a common response to these broad selective pressures. And when I say that Domatia have evolved multiple times, they have actually evolved an estimated 600 times in angiosperms, which is really crazy to think about. But if they have evolved so many times and are so highly convergent within angiosperms, this leads to the question, could other groups facing similar selective pressures also evolve this trait? And where else could this mutualism have evolved? So this is cinnamon fern. It is a really common species of fern that you have probably seen out in nature before. And if you were to take one of the fronds of the fern and lift it up, you would see these little tufts of hair that are located in between the rachis, the middle of the frond, and the pinnae coming out of that. And they're usually kind of white to cinnamon colored. And these tufts have been used by naturalists for a long time in order to identify this fern in the wild. However, a formal investigation of the function of these hairs has never been fully done. And it could be nothing, but when you look at the tufts of the cinnamon fern compared to the Domatia on angiosperms, you can't help but notice a similarity in terms of morphology and location on the plant. And that led me to my research question, do the tufts on cinnamon fern have an analogous function to the mite Domatia on angiosperms? To test this, I also had the privilege of going to the University of Michigan Biological Station to conduct this research. So what we did was we found a natural population of cinnamon fern on the property, and for each individual in the experiment, we pruned them till they only had 2 non-overlapping fronds. And on one frond, we covered these hair tufts with this tree pruning tar, making them inaccessible. And on the other frond, we left these tufts open and accessible, just applying a little bit of that tar to the adjacent pinnae to control for any effects of the chemicals. We left that on for 3 weeks. At the end of that, we pruned, collected the fronds, and then looked at them under the microscope to make note of mite abundance, richness, and number of Domatia. And what we found was really exciting. So the first kind of biggest result is that fronds with accessible Domatia had more mites. So you can see this pretty clearly here, that brown is the experimental group with the block Domatia had a significantly lower amount of mites compared to the fronds with open Domatia. And not only was there a greater mite abundance on the open fronds, there was also a greater mite richness. You can see there again in the green, the fronds with open Domatia had a greater number of mite morphospecies. Then finally, of the fronds that had open Domatia, we found that the number of Domatia was significantly positively correlated with the number of mites. We did not see this significant relationship with the blocked fronds, indicating that the abundance of mites was indeed due to the Domatia presence, not just the size of the frond. And then the final step is looking at the community composition of the mites that were on the ferns. So Domatia as this highly convergent trait are less defined by their evolutionary origin and more by their ecological function. So to strengthen this argument and this conclusion that these structures do indeed facilitate a mutualism, we would expect to see that the mites on our fronds are more predatory and fungivorous than they are herbivorous. And while in the field we counted morphospecies, however, I don't know if you've ever tried to identify little tiny mites running around by morphology alone, but it's quite hard, especially when you have nymphs and everything. So we are currently extracting DNA from the mites we collected in order to get a better sense of the species of mites that we are seeing. But we predict to see a greater number of predaceous and fungivorous mites compared to herbivorous. So what does this all mean? I first want to highlight that this is the first discovery of Domatia and the Domatia mite mutualism outside of angiosperms, which is really cool and opens the door to a lot more questions and research that can be done. I also, on the right there, that is an SEM photo of the cinnamon fern Domatia. It's pretty cool. Thank you, Rosie. This also means that these evolutionary patterns of defense are more generalizable than maybe previously believed when they were only in the angiosperm clade. It also means that Domatia and this Domatia-mite relationship is a reliable response to these broad, common selective pressures, even among distantly related lineages. It also means that Domatia could exist in a greater diversity of plants. We just haven't found them yet. This photo on the right here is of the hairy lip fern. I think there could be mites in there. And in terms of future work, ferns are really understudied, so I have a lot of questions. It wasn't until fairly recently that studies came out showing that not only do ferns experience similar levels of herbivory compared to angiosperms, but also that they do invest in defenses like volatile compounds, nectaries, and now Domatia. And I would love to expand this work to include a latitudinal, phylogenetic, and temporal analysis of the fern-mite relationship because I think there is a lot to be learned there. And I would just like to acknowledge Dr. Weber and the Weber Lab, the University of Michigan Biological Station for letting me do this research, and the EEB department in general for supporting me. Thank you.
All right. Anyone have any questions? There we go.
Hi, that was super interesting. On the ferns that you didn't block the Domatia on, what determines the number of Domatia? Is it basically related to the size, or are there other things also going on?
Yeah, it's pretty much just correlated to the size because for each pinnae, there's going to be 2 Domatia. So the larger and longer they are, the more Domatia they have. Yeah.
That was a great talk. Good job. I was wondering if you could talk a little bit about mites being specialists versus generalists, and what happens if your sequences come out in a clade of mites that are generalist and kind of do anything, or clades with just lots of variation in what they specialize on. Does that exist, or how much can you use the placement within the mite phylogeny to know something about their ecology?
Yeah, I think Generally, the mites that I showed here, those are families that are really common in a study in this similar area. About like 80% of the mites are going to belong to these two families, which the first family is mostly predaceous. Second family, there's some predaceous, some fungivorous. So I think for the most part, we should be able to tell their ecology.
Gabby, you're up.
Can you guys hear me? Is it working? Okay, wonderful. All right. Welcome to my talk. We're going to talk a lot about stomata today. Stomata in the field, opening up the mysteries of auxinic herbicide impact on stomatal formation, development, and function. I am in the Baucom lab. Shout out to the Baucom lab. Thank you guys, everyone who's here from the lab today. I'm very excited to be presenting some of the stuff we've been doing. So a little bit of background. What are stomata? Many of you might have heard of it. They are the mouths of the plant. Basically, they open and close depending on environmental factors. They're a big part of the facilitation of bringing in CO2 and also preventing water loss. So they strike that careful balance between being open and letting in CO2 for photosynthesis and not releasing water vapor or bringing in water vapor depending on the circumstances. They also play a big role in preventing pathogens from coming into the plant, specifically bacteria. Bacteria need stomata. They need some avenue to enter into the plant interior. Stomata tend to be the way to do that, and that's a whole other area of research that is super interesting. But we're going to talk about what impacts stomata. Mostly interferences with the complex systems that create the division of stomata. So stomata start off as a meristem cell. It's kind of like a stem cell of the plant. And they go through this long process. They divide symmetrically, a whole lot of steps. And anything that gets in the way of that can really impact how those stomata develop. There's also environmental conditions, weather, temperature, and chemicals, which is what we're going to talk about when we talk about auxinic herbicides. So what are auxinic herbicides? They mimic the natural structure of organic auxin, but they do persist longer. So auxin is a growth hormone in plants, and it's really good, and the plant produces it naturally. However, they figured out in the 1940s how to make a synthetic type that basically, instead of just signaling and then breaking down, it signals and then continues to signal. And that can be really detrimental to plants, which is exactly why it was designed. It was designed to kill weeds in agriculture. It works very well. But our issue is that some of it, about 1 to 10% of the herbicide, drifts onto natural populations or wild populations of plants outside of the agricultural space. So our research questions. Does auxinic herbicide drift impact the formation, distribution, and function of stomata in weed species? And what are the broader implications of herbicide drift on ecological interactions of weed species? So a little bit about the field work that we've been doing. I've been here since August during that summer field season. Shout out to everybody who was working out in the fields then. We did an experiment. So we had our 6 plots. You could see a little version of it in the middle. We had 3 trial groups, one with dicamba, one with 2,4-D. Those are 2 different auxinic herbicides, and then 1 water control. They were randomly placed within those 6 plots. And my data collection, specifically in terms of stomata, we did randomized leaf collection, 10% of a bunch of the plants of the 8 species, 3 of which being legumes, which we mostly wanted to look at, but we took general data just to see what patterns we could find. So here are some pictures I took on the SEM microscope. Those are super duper zoomed in. So some things that I'm looking at are stomata density. Stomata have a certain density. They have a certain spread outness that needs to be followed in order for efficient bringing in of CO2 and also for efficient opening and closing abilities. There's a lot of genes like the too many mouths locus. I love that name because it's actually, they create too many mouths. It creates too many of the stomata. It limits the efficiency of the plant. And so that's something I've also been looking at. We're also looking at stomata aperture. So how much they're open versus not open, and stomata open and close normally throughout the day and throughout the season. However, auxinic herbicides have been tested to actually just close the stomata, which is part of what kills those weed plants that it's applied to. We also have stomatal abnormalities, which I'll show on the next slide. There's some spacing abnormalities, and there's also some misformed individual stomata. Here's some of the abnormalities you could see here. This one is like too large. If you look at it compared to all the other ones, that means that something went wrong in the formation process. And these ones here only have one guard cell. So if you look at all the other ones, they kind of have two lips, like a mouth. These ones only have one. That means that something in that process did not go correctly. And those are some more examples of some of the morphologic phenotype traits that we've been looking at and trying to identify in our photos. We've also been looking at some leaf scans. So we're assessing herbivory and pathogen damage, looking at different amounts of mildew and how much of the plant has been taken out. If you look in the first picture, you see they're pretty good looking leaves. They got a little herbivory damage, but they're generally doing well. Over here, not so much. We got some brown mildew, we got some white powdery mildew if you look closely, and quite a bit of herbivory. So we're sort of also at the same time looking at what might be other effects of this auxinic herbicide and maybe where can we apply the stomata research into that. Here's a little bit of some of the data analysis we've been doing. We have our 3 treatment groups. This is just sort of an initial looking at the data. We have 2,4-D, control, dicamba. These are our 3 legume species that are sort of the center of our research, but we did look at 8 species across. You can see some maybe differences among the groups. Again, we have to do more tests, but this is sort of a good look at what we've been seeing so far, and wanted to show you guys sort of the initial look at stomatal density by treatment and by plant species. So for next steps, we're just sort of beginning. Our field season just ended in mid-October, so we're really starting right now to get to the next part, investigate the symbiotic relationships. We're looking at the root system, specifically of those legume species, see how much the CO2 brought in by the stomata might be affecting how many rhizobia there are, how many colonies, how many root nodules. We're also analyzing root samples, like I said. We're going to run more statistical analyses to get more of those pretty graphs like we saw in the last slide. And we're also going to be collecting more data on stomatal characteristics. So there are a lot of things to look at, and you can see kind of the variety of stomata in the species that we have right over here. Any questions? Thank you, guys.
So you were on the last slide, you're talking about looking at like nodulation and stomatal density.
Yeah.
But wouldn't number of leaves maybe better predictor of nodulation? Or do you have counts of number of leaves?
Yeah, we have that
biomass
data, but we're also looking, my sort of thought was if you're not having efficient bringing in of CO2, there's less of a mutualistic relationship potentially because
100%. I do not disagree with
100%.
But I was just wondering if the size of the plant could also play a role in, you might, like if you corrected for the size of your plant, it might be a better indicator because you can have less efficient stomata on many leaves.
So yeah, we definitely have like leaf size data, and that's why we took those scans over here.
Mm-hmm.
We're going to start measuring those on ImageJ, get some more data. We took about 10% of each plant, so it would be consistent and we wouldn't have to kill the entire plant. But yes, I definitely agree.
Yeah. Do you have plant height?
Do we have plant height? Vero, do we have plant height? Shoot biomass. We have shoot biomass.
I use that. Very, very cool.
Thank you.
All right, time for one more.
Super cool that you're looking across a bunch of different species and that you might be seeing sort of differential effects of your treatment on the species. What are the traits of those plants do you think might be driving the different treatment effects that you're seeing?
That's a great question. And I think that's something we're looking into. Do you mean like physical traits?
Yeah, or like you obviously have some variation in the photos at the end you showed like different densities and as Amy mentioned, all your plants look slightly differently. So if either physical traits or growth traits or physiological traits or
Yeah, so like within like these scans, we have here some of the stomata are a little bit more buried. Can't really see in this one, but they're more buried in between the guard cells. So we're definitely taking that into account, but also within a plant, there are differences in stomatal morphology, which I didn't realize until I started taking a look at the pictures, but I was like, wow, these were all taken on the abaxial sides, the back side of the leaf. And still there's so much variation. And sometimes I look at two pictures and I'm like, those are the same species, that's crazy. But yeah, there's definitely stuff to consider there. I've been categorizing the stomata to sort of see like some might look more abnormal because of the original intent of like their processes and what they originally look like. But yeah, I'm definitely taking that into consideration within the context of the stomata. Yeah, amazing. Thank you guys.
All right, Stella.
Okay, can everyone hear me? Is it working? Good. Up a little bit. Is that better? Better. Okay. I've never used one of these. What button do I? Oh, it's that. Okay, got it. Okay. Hi, everyone. My name is Stella. I'm a senior working in the Coastal Ecology and Conservation Lab. And today I'm going to be talking about work that I've been doing in the invertebrate sector of this lab and how invertebrate communities are differing across different artificial reef habitats and the work that I've done the past 3 years within this lab with the sample processing. And now that I'm starting to do my thesis, what my plans are for that. So first, I just wanted to give a little bit of general background about coral reefs. So as we all know, coral reefs have been increasingly threatened by different, a wide range of a wide range of stressors such as climate change, overfishing, pollution, degradation, which is driven by tourism. And these pressures are not only harming the coral species specifically, but also all the different organisms and ecosystems that rely on the reefs for habitat, food, and nursery grounds. In addition to this, in many tropical coastal regions, these reef ecosystems serve as the primary source of protein for the people living there. So further emphasizing the need to maintain the ecological health of these reefs. And mitigating these threats is critical to preserve the ecosystem services that the coral reefs are providing for both the animals and the humans relying on them. So one promising strategy for coral reef conservation is the use of artificial reefs, or ARs, which is kind of covered by fish there, but in that photo. And kind of a general definition of an AR is a man-made structure composed of any type of material, concrete, steel, rocks, anything that you could throw in the ocean, basically. And that's specifically designed to mimic the physical and ecological functions of a natural coral reef. So a little bit about more specific to our lab, these reefs really function as ecological surrogates for a natural reef, and it has been found that ARs can provide this hard substrate, which facilitates the colonization of sessile invertebrates, such as coral or sponges or any invertebrate that's sticking in one place. And by increasing the habitat complexity of these reefs, the ARs can boost species richness and abundance of these sessile organisms, offering these shelters and breeding grounds and foraging opportunities for these individuals. Studies have also looked into that over time, like there seems to be an increase of abundance in all types of invertebrates, not just sessile, but it is something that's less studied overall. So that kind of brings us into a super general overall question of our lab, or my section of the lab, that is like, how are these artificial reefs impacting invertebrate populations? So this is our study site. It's in the Bight of Old Robinson on Great Abaco Island in the Bahamas. This is the bite right here. Oh, I guess I have a clicker. This is the bite. And then there's, oh my gosh. And there's a bunch of different reefs all throughout this bite. So a little bit into the field sampling. I didn't participate in this, but I think it's important to go over because it kind of helps you understand the sample processing, which I'll go into next. So there are 20-meter transects that extend on the north side of every AR that we study for the invertebrates. I believe there were 5. That's what the AR looks like. And you take they took samples at 1, 2, 3, 4, 6, 8, 12, and 20 meters and these 0.34 meter square plots. And they were dredged basically using a vacuum-like tool where it just sucked all of the detritus and mass and whatever was on the surface of the seafloor in these little plots. And then they were bagged and frozen and brought back to the lab. And so a little bit into what I did these past couple of years and a bunch of other undergraduates as well is the samples were first thawed and then they were washed through decreasing mesh size sieves. So if you're unfamiliar with what a sieve is, basically I like to picture like a window screen, and then they were from 12.5 millimeters all the way to 0.5 millimeters. So the samples were just washed through in order to separate it and make it a lot easier to identify invertebrates and actually count them and sort them. So after we did that, they were identified either using a microscope or the naked eye. You could really only see until 2.8 millimeters with the naked eye. These are some of the biggest samples we found, that crab and this gastropod. Pretty cool looking. And then the other ones are examples of what you would see under the microscope. And we identified them into 4 major groups: bivalves, crustaceans, gastropods, and others. And then the bigger samples, of course, if you could see them and identify them further, we would to get more accurate data. So overall, we counted and then measured over 27,000 invertebrates, which was a lot. It took us a long time. Finally finished that. After we counted them, we also freeze-dried them, weighed them, incinerated them, and weighed them again, every single one of them, in order to get biomass and then therefore get production levels. So it took us a while. I'm glad we're finally done with that. So that brings us into what my thesis is going to focus on. And I want to look at how this invertebrate biomass and production is varying across artificial reefs in relation to seagrass production. So we have seagrass biomass and production levels already for the entire bite, like all the reefs that are in the bite, but we only have invertebrate for 5 of those reefs. So the plan is to create correlations and regressions to be able to really understand the invertebrate production and biomass across the entire bite. And then second to that, I want to create a trophic pyramid to visualize the production of the whole system, including the fish as well, and get really this overall understanding of production levels in artificial reef ecosystems. So next steps, first would be those correlations that I just mentioned. That was a very preliminary plot that I made, so don't look too closely, but that is what they will look like. And there is a small positive correlation between seagrass blade height and invertebrate production. So we're going to use that and create regressions to scale that across the entire bite. And then that brings us to the scaling. We need to design a method to figure out production everywhere because we couldn't measure every single seagrass blade height across all the reefs. So we have to figure out a good way to accurately extrapolate that data and make sure we're having accurate predictions of the production of invertebrates. And then finally, we have production for seagrass, our primary producers, and we do have production for fish, which is our secondary consumers. So the goal of my project is to fill in this missing piece, which is our invertebrates, and figure out production there and trying to identify the whole energy flow of the entire reef. So that really brings me into why this work matters. So invertebrates are one of the foundations for how these artificial reefs are functioning, and they're really driving nutrient cycling and providing energy that the higher trophic levels, like I mentioned on the last slide, are relying on. So by looking specifically at production and not just at abundance, we're getting a clear picture of how the reef is actually functioning. And because we still don't understand all the ecological processes, such as production of seagrass, that shape the artificial reef's performance, this can really help fill that knowledge gap and bring it all together, which leads us to how this really is really showing us the bigger picture of the entire ecosystem and how we can, it's not just showing us what specific species are there and how many individuals are there, but how much energy and productivity the entire system is supporting. And by understanding that, we can really support more informed decisions about effective conservation and help support fisheries in this area that rely on these types of reefs and this type of ecosystem for their primary food sources. So then my acknowledgements, obviously everyone in the Coastal Ecology and Conservation Lab that has helped me out, Nepsis for signing me up for this, Jake for, I just pressed the button, Jake for helping me out with my thesis and getting me to where I am now, and then Shreyan and Mike, who are no longer in the lab, but were in the lab last year and have done a ton of work for this invertebrate project and gotten it to where it is now. Thank you. Any
Love the invertebrate focus. I'm curious, and sorry if I missed this, but do you have a sense of what the invertebrate communities are like on non-artificial reefs? And like if you could go back 100 years to a healthy reef in this system, like what the invertebrates might be like? Are there like museum specimens or old papers or something?
I'm sure there is. Yes, it's not something I've 100% looked into yet. I'm very much in the preliminary stages of my thesis right now. So that's definitely something I want to do. It's like, I think necessary to compare it because then we really know if these reefs are functioning in a positive way. So that's something we will have to do. I just haven't gotten there yet, unfortunately.
Cool, thanks.
I suggest when you start taking your data that you separate out the difference between those that have mineralized tissues and those that only have soft tissues, because then you'll be able to compare your results to all the coral reef data from the fossil record, which goes back tens of millions of years.
Thank you.
That feels like a mic drop.
Thank you.
That brings us to our last speaker, Drew. I just want to point out that all of our speakers so far have stuck exactly to their 10-minute limit, which if you've ever been to a conference, you know is not usually the case. So thank you all. Take it away.
Cool. Okay, can everyone hear me? Sweet. Oh, that's the wrong way. Sorry. So I'm Drew. I work in the Allison Davis Roboski's lab, and I'm going to be presenting on functional accommodation in extreme venom glands of Cossus snakes. So when most people think snakes, one of the first things a lot of people think about is venom, right? It's kind of scary. It's a little bit of the way they're portrayed in media is based on that. But how does that actually work? So the venom gland is pictured here in green. This is Echis carinatus, which is a member of the viper group. So if you think like rattlesnakes, that's part of that group. And this represents kind of a standard morphology that you would see. So you see this like duct attaching to the enlarged tooth here. And then you have kind of the main venom gland. But what my research is focusing on is a really kind of derived morphology within Viperidae. And that is the genus Cossus. So this is still a venom gland, this green structure here. And as you can see, it's way different, right? It's massive. It elongates up to about a third of the body length. And there's so many other weird things about it too, right? So this actually, you'll notice it's on top of the rib cage, right? So most organs, most viscera, you would expect they'd be in the body cavity. But actually this is deep to the skin, but lateral to the body cavity and to the rib cage. And Cossus presents kind of a really unique opportunity, in my opinion, for studying the evolution of novelty, right? A lot of times kind of novel structures can take millions of years to develop. And so that forces you to go look at the fossil record. And if we know anything about fossil data, it's that it's often incomplete or only focuses on mineralized structures, right? And so Cossus is really unique because they're pretty based on modern molecular phylogenies. There's like a couple, I can't think of the names. Obviously, Dan's paper with title, they did, that's the phylogeny we're using for this figure. But you can see Cossus is pretty well nested within the old world viper clade. It's not like a Xemiops, right, where it's on its own kind of branch. And then actually even further than that, there are other species of Cossus that do not have an elongated venom gland. So we can see Cossus diphilippi here, right? The venom gland it's definitely derived from kind of our outgroup Echis carinatus, but it is still contained within the head as compared to this monstrosity that a lot of the other species have, right? And so, like I said, there's kind of a lot of different angles we could take looking at novelty here. But one of the questions that I think I want to walk you guys through today is looking at homology. Specifically, we're looking at the homology of the adductor mandibulae externus superficialis. We're going to call it AMES, so I don't have to say that a billion times. But that's that pink muscle here. It's an adductor muscle, right? So it attaches to, or sorry, originates on the quadrate and inserts on the compound bone, the large part of the mandible, and it closes the jaw most of the time. And in Cossus dephilippi, right, we see kind of it's almost like being invaded by the venom gland here. And then in Cossus racemus, we see these structures that are attaching to the AMES, right? And so one of the central questions that we had is, what are the homologs of these slips here, right? Because these go all the way. It doesn't. This is a head scan or of the Cossus, so you can't see the rest of the venom gland. But these strings of muscle, these slips of muscle, do go all the way back to that extreme length we saw earlier. And so, kind of the central question is like, the fiber direction and the length is so different from what you would see in the normal AMES morphology, right? So our two kind of working hypotheses are, one, it is still the AMES, but it's just been grown alongside the venom gland, or it could be kind of a co-opted epaxial muscle. So epaxial muscles, think like the longissimus, right? They're lining your vertebrae. And so just kind of like based on the evidence that we have, we are going to suggest, or right now, tentatively, that these muscle slips are derived from the AMES and not derived epaxial musculature. The first piece of evidence for that is that the epaxial musculature in long-glanded Cossus are pretty much the same as other vipers and short-glanded Cossus, which kind of suggests that you would have to not only evolve this long venom gland musculature, but then also modify your existing epaxial musculature, right? So that's kind of less parsimonious than suggesting one modification, kind of at a non-parametric level anyway. But then also snakes kind of have this weird genetic architecture that allows them to do a lot of elongation, right? So if you look at the vertebrae count in snakes, they have a ton of vertebrae. But like giraffes, for example, have similar or same number of vertebrae as humans, right? So this kind of like copy-paste mechanism might actually be pretty I guess not ubiquitous, but it might be easier for snakes specifically to elongate organs. And we see that also in their liver and their heart. So those are kind of two pieces of evidence that we're working with. And then also, I just kind of want to wrap up by talking about, the mechanisms behind this. Obviously, this is all morphological data, right? So we can't say the exact mechanism of how this is working, whether it's co-opted genes that already exist and are being expressed differently. But also could be integration, right? So developmental integration, oftentimes you have, if you have stuff from the same germ layer, it kind of depends on each other for development, right? So if you think skull structures, so maybe that's what's happening here, right? We have a venom gland that has modified genes for hypertrophy, and then whatever is associated with it might also have to accommodate that developmentally, right? So we're not saying anything about the mechanism because we don't have data on that, but I just think that Cossus is a super unique model for looking at those kind of questions that I think are pretty central to evolutionary biology, right? Because novelty is kind of the smaller scale version of how do we get the diversity that we see on Earth. So yeah, and I just wanted to thank my lab as well. You guys have been awesome. And yeah, if you guys have any questions, happy to answer them. So yeah.
Snakes are cool.
Yeah, snakes are cool.
It's a really cool guy. So I know nothing about snakes, and I don't know how realistic this is. I don't know if the lab happens to have just a batch of them, but my immediate thought is just, how does this occur developmentally? Is the gland always proportional, or does it grow later? And how realistic is it to grow a bunch of baby snakes and then, I don't know, CT scan them away?
That's a great question. I think So first of all, I want to shout out the UMMZ because that's what makes all of this possible. These CT scans are all from dead museum specimens, not actually living causus. And so we don't have a lot of good access to growing embryos and stuff. There has been one study on the development of elongated venom glands, but only in one species of causus as well. And it found they were like fairly early developing. So it's not like that was one of my first questions is like, oh, like, normally when you see, or not normally, but oftentimes when you see crazy structures, it has something to do with like sexual dimorphism or competition. But it shows up, as far as we can tell, really, really early on in development and in both sexes, although the data on that is super low. So don't quote me on that. But yeah, no, I think it's super interesting. We just don't have much data on it, unfortunately.
This is for all the speakers. Thank you so much for doing this. This was so cool. I know it was probably nerve-wracking, but these were so impressive. And it just reminds me again that this is an amazing, amazing department where we get science presented by undergrads at such a high level. And then so many people turn out to see it. I mean, there are faculty and postdocs and grad students and staff here to support you all. And this is unique. I don't know of other departments that are like this. And so thank you all for doing this. And thank you to your mentors for mentoring you. So how about a round of applause? I see it starting right there.
To add on to that, we would love to do this again next semester. So PIs, if you have some undergrads who you think might be ready to talk, not just PIs, grad students, postdocs. If you have undergrads you think would like to talk next semester, let us know. And we'd be happy to put another one of these together if we have enough interest. So thank you all.
Thank you. Thanks. Thank you.
I have my like extra back. Bye.